Hi, do you see me?
Yes.
Yes, yes, yes. And here, yes. Mm-hmm. Now, why it is not you are presenting to everyone? Why I don't see? Yes. Now, please. I receive a lot of mails from you about our midterm one, and what are you interested in? Please ask me. Are you here?
Yes.
Yes. And after next week, we have today's lecture is dentin, and one more lecture will be dental pulp. Yes. After next week, you have the midterm one. Yes, I sent you this from previous topics. I sent you the questions, and on this weekend, I will send you the questions for dentin and questions for dental pulp. Yes, and these
You can
But
study
where are the
these questions.
answers?
will be included. Yes,
Will the
I'm here.
are the exam is the exam going to be like from these questions, the same as these questions, or similar
They
to them?
let me say similar
Yes, because
questions.
the doctor like too many packed information and the
Many
book.
information, yes.
Yeah, too
Then
many
no,
information.
I'll try to. You have 40
But where are
questions,
the
yes?
answers?
You have 40 questions, yes?
Who knows the answers of
Yes, 40
the
questions for tooth morphology, then
morphology?
animal
Anim
morphology or
Shad?
structure, dentin and dental pulp. Will be
Mm,
about 100 questions. Yes, I try to. There are 30 questions.
The exam is going to be
There
only
are
30
30
questions.
scores. 30 scores for each midterm. You have 30 scores, and for final exam 40.
So the exam is going to be 30 questions.
30 questions.
The 30 questions are from the 100.
From 100 is one third.
but where are the answers?
Hmm?
انت بدك
Where are
you
the
should you
answers?
should answer them.
You should know the answer.
Okay, but
You
we should be
you
sure
already
from
did.
our answer.
خلاص على مشاك مسلسل.
You
You
said
have
that you know
answers.
answer.
You have questions. Would you like answers?
Okay, so we should find the answers, right?
لا ما
I
انت
don't.
بتعرفهم هلا كنت عم تقول انت بتعرفهم.
Mhm. Let's. Any questions?
Yes, yes. The 30 questions in the midterm one will be from the 100 questions you will give us.
The questions will be from tooth morphology because the first lecture, the overview of oral cavity, with this, for example, oral mucosa. Yes, we discuss it is certain topic. We discuss it with to the lecture, and only tooth morphology. It is the second chapter, dental osseous, second chapter, and there are the description of maxilla and the mandible and tooth
Yes, yes.
morphology.
I know these information,
Yes.
but my question is that you will give us in total 100 questions to solve, right?
Yes, less than from these 100 questions will be 30 questions from these questions which you have, and I'll send in future. I send on dentin questions and dental pulp. These two topics will be involved in this midterm one.
Okay. Then
Yes,
the midterm exam will be 30 questions from the 100 questions that you will be sending us by the next week.
yes.
Okay. Thank you very much.
Hmm. Let's let's begin now. Today's our lecture to topic is dentin. You know that dentin is one of the mineralized structures of tooth. There is, we already discussed enamel, and dentin and cementum. These three parts of tooth are very heavily mineralized parts, but the hardest and the sturdiest material is enamel, enamel, and the combination of enamel and dentin provides a rigid, hard structure suitable for tearing and chewing that persists both abrasion and fracture. The cementum, you know that dentin underlies the enamel, and in the root, cementum covers the dentin of the root and anchors the tooth to the bone of socket via the periodontal ligament. The junctions of dentin with these two other hard tissues are biologically unique as in the tissue itself. These two major properties, there are two major properties that distinguish dentin from enamel. You know that enamel is the mostly mineralized tissue. 97% there is mineral material, and only 2% is organic material, and thereby dentin, which contains 70% of inorganic material and 20% organic material. There are two major properties which distinguish dentin from enamel. That, firstly, dentin is sensitive. And secondly, dentin is formed throughout life. You know that enamel after eruption is it has a node, these cells which produce in enamel, and they are produced until eruption. But dentin is formed throughout life, increasing in thickness at the expense of the dental pulp. You know that dentin is surrounds here, you can see dentin surrounds dental pulp. This is dental pulp, chamber of dental pulp, and the dentin surrounds dental pulp. And this is reflected in the presence of unmineralized layer of dentin matrix at the pulpal surface. And this is predentin, and we discuss it a bit later. The physical properties of enamel and the dentin are complementary, and thus, although enamel is extremely hard, it is unyielding and
Uncle,
may fracture.
question is,
Yes,
uh,
do you hear?
yes, but you are presenting or not yet? We can't see
Yes,
your screen.
do you see the presentation?
No,
No? Oh, excuse me.
حبيبتي ولا يهمك. We stopped presentation.
Mhm.
Presenting.
Do you see?
No.
No, excuse me.
Yes, now.
Yes?
Yes, now it's okay.
Yes, do you see? And here.
Yes,
Yes,
thank
yes,
you.
you can see it is dentin. It is dentin, and it surrounds the dental pulp, the chamber of dental pulp. Yes, and here you can see this is the junction of dentin and enamel, and on in the root there is junction between cementum and the dentin. Yes. And together, dentin and enamel is very rigid structure, a hard structure, and it is suitable for chewing and fracturing. And at the same time, dentin is more flexible, more elastic than enamel. Dentin is derived from dental pulp, dental pulp. The cells which produce dentin are in the periphery of dental pulp, and they are called odontoblast. And we discuss it with dental pulp and with odontogenesis and development of tooth. Now, uh, here you can see this is dentin and surrounding enamel outside in the crown. Fresh dentin is pale yellow in color and contributes to the appearance of the tooth through the translucent enamel. Dentin is harder than bone and cementum but softer than enamel. Dentin is much more resistant to propagation of cracks than enamel. Higher fracture toughness. It has higher fracture toughness because of the intimate association of smooth apatite crystals with strong protein fibers. You know that in a dentin, these apatite crystals are embedded in collagen fibers, and this stabilizes this inorganic content of dentin. Then dentin, its organic matrix and tubular architecture provides it with greater compressive, tensile, and flexural strength than enamel. Dentin is permeable. The permeability depends on the size of patents and patency of the tubules. You know that these cells, odontoblasts, and we'll see it soon, these odontoblasts, they have processes, and through these processes run throughout the whole thickness of dentin in the special spaces which are called the dentinal tubules. And this amount of tubules and processes will decline with age. Cracking occurs in dentin when it has been weakened by caries or cavity preparation. Then, what organic matrix contain the main part of organic structure of dentin? There are dentin like cementum, and bone is composite material consisting of apatite crystals on the organic scaffold, predominantly composed of collagen. And this collagen is collagen type 1, and the dentin, approximately 70% inorganic, 20% organic material, and 10% of water by weight, and 50% inorganic, 25% here you can see, 25% organic, and 25% by volume. The inorganic mineral component is in the form of calcium hydroxyapatite crystals, and these crystallites are calcium poor but carbonate rich in comparison with poor hydroxyapatite. And although similar in shape, they are smaller than in enamel hydroxyapatite crystals. Then,
Doctor, we
organic
can't see
matrix.
the screen.
Yes,
We can't see the screen, the
don't
PowerPoint.
see?
Yes, we
بلا
can't.
بلا.
ما أنا مبين هون.
Do you see or not?
بص على أنا أجلادزي.
خير بس خير.
Yes, it's my name, Anna Agladze. Yes, do you see the
Okay, no
presentation?
problem, no problem.
Yes, yes, yes.
Yes. Yes, then organic matrix of dentin, which is crystallites, are these embedded in, as we say, is embedded in the organic matrix, and this is collagenous, and this collagen assume this plasticity to the dentin, and it consists of fibrils embedded in amorphous ground substance, And collagen fibers, they consist about 90% of all organic matrix, and other organic matrix may be traced, such collagen type 3, collagen type 5. And in here you can see on this graph, here you can see collagen fibers. These collagen fibers in predentin, this is predentin, this is dentin, and this is odontoblast zone, and this is intercellular fibers here. And these collagen fibers in dentin, you can see that they are larger, 100 nanometer, and are more closely packed in the predentin. They are parallel to the pulp, the dental pulp cavity. Collagen fibrils in dentin are not assembled into bundles as they are in many non-mineralized connective tissue. And also, alongside with collagen fibers, there are other non-collagenous proteins, such about 8%, such as there are glycoproteins, proteoglycans, phosphoproteins, acid proteins, and growth factors. These all of these proteins are involved in mineralization. You know that this predentin is hypomineralized. It is newly formed, newly produced dentin, and it is not mineralized. And after mineralization, they assume such structure. The fibers are more larger, and they are very tightly knit to each other, intermingled to each other. Yes. Then mm, about dentin growth factors. There are some growth factors which are very important in mineralization, such as dentin matrix protein 1. It presents in dentin and bone and is thought to play a crucial role in mineralization, and it can initiate apatite nucleation. Also, there are a lot of phosphoproteins which are very important also in mineralization process, because they have a lot of phosphate group and carboxyl groups, and they are negatively charged, and they bind calcium ions and play important role in in mineralization. This dentin matrix protein 1, dentin matrix protein, is very important. It is in small amounts in predentin here and intertubular dentin, but is strongly represent in peritubular dentin. And in the experiments with the mice, null mice show abnormalities in mineralization process. There are also other proteoglycans, such as non-collagenous proteins in dentin, and they are such as biglycan and decorin. Also, glycosaminoglycans are primarily chondroitin 4 sulfate, chondroitin 6 sulfate, and they are their role in collagen fiber assembly. You know that they participate in this process, and their cell mediated effects such as cell adhesion, migration, proliferation, and differentiation is also very significant. There are also growth factors found here, such as insulin growth factor, bone morphogenic factor, and transforming growth factors. And also, in a small amount, you can find that there are 2% of organic content of dentin, such as lipids, and these lipids play a role in mineralization. They are in the form of phospholipids, of cholesterol, and phospholipids have been detected in both predentin and mineralized dentin. Now, here you can see these cells. These cells are odontoblasts. Here schematically diagram shows these odontoblasts, which are peripheral row of dental pulp. These odontoblasts. Odontoblasts are polarized cells. They have such long processes. These processes, they are specialized for collagen and glycosamine synthesis and are bound together by junctional complexes, these odontoblasts. And they have no basal lamina, and the collagen-rich matrix called the predentin is formed here. It is first layer of dentin production, predentin, and there are the different types of dentin and odontoblast structure we discussed on next lecture with dental pulp. Now, you can see also, you can say that they have such long processes which are located in special tubules, special spaces, which are called tubules, and these tubules are located through all thickness of dentin. Here you can see these are tubules, and here you can I'll show you the here you can see these are tubules, this is pulp, periphery of pulp. These cells are odontoblast cells, and you can see these odontoblast processes. And here, this is transverse cut, and here you can see these holes which are odontoblast processes. And here you can see this is schematically these cells which are odontoblasts, and they have such processes and dentinal tubules, and these dentinal tubules are filled with fluid, and also you can see that nerve fibers are encircle these cells and their processes. These are tubules, and they penetrate the whole thickness of dentin. Here you can see this is tubule, and you can see this process inside the tubule. And here you can see this is scanning electromyograph, and you can see deep dentin displaying on odontoblast, and this is odontoblast process in dentinal tubule. Now, these tubules taper. Here you can see that they taper from approximately 2.5 micrometer in diameter and at their pulpal surface, and they at their ends they can be 1 micrometer or less at the periphery. During the formation of dentin tubules by the odontoblast, the cells migrate toward the occupy smaller surfaces. They move, and the tubules are more widely separated at their peripheries. Approximately 22% of the cross section of these tubules they maintain these processes, but due to aging, this amount of processes is decreased in the tubules, and at the junction, enamel dentin junction, there is only 2 or 3% with processes, tubules with processes. Now, the tubules also show changes in direction in a much smaller, you can see this here. Here, these processes they have such curvature, which is called the primary curvature. primary curvature, and they have such sigmoid structure, and I try to show the other picture. No, this is primary curvature. Yes. Mhm. In their convexity in the primary curvature is nearest to the pulp, and then this convexity it is to rootward. Yes, in the root and beneath the cusp, the primary curvature are less pronounced. At the same time, there is the other curvature, which is called secondary curvature, and in some here you can see such deviation from the line, and they are wrinkles, such wrinkles, which is called secondary curvature. And I'll show you the picture now. And at low magnification, it gives the appearance of a line crossing the dentine. Here you can see the here this here crossing dentines. It is a micrograph of transversely sectioned dentinal tubules. You can see these tubules, and these lines of secondary curvature, which is deviation from. I'll show you this. Where is this secondary curvature? In a moment. Oh, this. Here you can see this is primary curvature in the crown, and in the root you can see that it is not so pronounced as in the crown, primary curvature. And also you can see on these lines, when these lines have such deviation from and amplitude about 30 micrometer or 40 here, for example, you can see these wrinkles of these lines, and they are called secondary curvature. Yes, and they are called the contour lines of Owen. These are not commonly seen in most of the dentine, but usually they are evident in the junction of primary and secondary dentine. You can see that these are dentinal tubules. Dentinal tubules. This is longitudinal cut, and here you can see these are tubules, and here you can see these transversely cut structures. Here you can see this is dentinal tubules, and these are dentinal tubules. These tubules, between them there is mineralized intertubular dentin, mineralized here around these dentinal tubules, areas called intertubular lines. But beside this intertubular mineralized collagenous dentin, inside the dentin you can see the peritubular or intratubular dentin, and here you can see this is tubule, and this is intertubular dentin, collagenous dentin, and you can see this difference between the sides of tubules, and the sides of tubules are peritubular or intratubular dentin. This type of dentin is known as you know, intratubular dentins, and this dentin differs from intertubular dentin in lacking of collagenous structure. This also relates to the findings that it exhibits other physical properties than intertubular dentin. It is rich in glutamic acid and serine. It lacks dentin, phosphorin, and other rich in DMP1 or dentin morphogenic factor, and together with its lipid content, its composition has been classified as proteolipid phospholipid complex. It differs from intertubular dentin, and here you can see these lines on sides of tubules, and these are intertubular dentin. This A shows the diameter of tubules, and inside this line you can see intertubular or peritubular dentin, which differs from in intertubular dentin, which is more collagenous, and this intratubular dentin is contains more lipids. It is proteolipid, phospholipid. It has proteolipid, phospholipid composition. And here you can see these are tubules, lumens of tubules, and you can see here peritubular dentin on sides of these tubules. And here you can compare how intertubular dentin, these are collagen fibers, collagenous structure, and it differs from here, from surrounding peritubular dentin in the tubules. Now, now let's speak about this. What is this? It is translucent dentin, and this translucent dentin is because these dentinal tubules, they can be occluded, and their composition, their refractive index is equal, similar to the intertubular dentin, and they have such appearance, translucent appearance. But it appears with aging in sclerotic dentin, and you can see on this transverse cut how these dentinal tubules, how they are diminished and occluded, almost occluded in these tubules. And the right side is the occluded tubules, and their refractive, when they are in water, their refractive index is different from intertubular refractive dentins, the refractive index, and they are such opaque. Now, here you can see these tubules, and you can see this is predentin, and you can see this is mineralizing front, and you can see dentins, which is filling with hydroxyapatite crystals. And this is tubule in this micrograph, and you can see this odontoblast process, and from both sides you can see this fluid in the this tubule. There is fluid, as you know, in the tubule, and odontoblast process is in this tubule, and it receives nutrition from this fluid, and this fluid is derived from dental pulp. Now, here we can discuss the composition of these tubules, and you can see that in the tubules there is the fluid, which contains nutrients for odontoblast processes. Also, it contains such nerve endings. You can see these nerve endings, and it may be this structure, this spaces is called periodontoblastic space, and sometimes with the aging, this process retract, and there may be the space which is called post-odontoblastic space, and these spaces are filled of this fluid. This is extracellular dentinal fluid, the precise composition of which is unknown, but it is thought that they contain nutrients and other substances which are important for odontoblast processes life. Then um, you can see that the cell body of the odontoblast we study with dental pulp, and there are these processes, and they contain such nerve endings, and the nerve endings you can see that it contains microtubules and mitochondria. It is unmyelinated nerve ending, and the mitochondria are sometimes present here, and the microtubules and intermediate filaments run longitudinally, and in the predentin and the very innermost mineralized circumpulpal dentin, the odontoblast processes seems to occupy the full width of the dentinal tubule. Yes, at these levels, afferent nerve axons. Afferent nerve axons are also seen here. You can see this odontoblast process and afferent nerve axons, which contain, you can see, a lot of mitochondria and also microtubules and fine filaments. Then, mm, there are some hypotheses how these axons are they propagate, they generate the nerve impulse, and the most plausible hypothesis is that in this fluid which is in odontoblast tubules, they contain potassium ions, much more than sodium ions, and this is the source of polarization, but the polarization way which then irritates nerve impulse. Now, terminal branching of tubules. You can see this terminal branching of tubules at the enamel dentinal junction, and they are very widely spaced in the dentin. These tubules can unite with other tubules branches, and this is at the dentinal junction, and thereby dentinal junction, enamel dentinal junction has such wavy appearance. Now let's describe the different types of dentin, which is due to their composition of and deposition. Peripheral to the predentin, where is here? Peripheral to the dentin, this is called mantle dentin, and the mantle dentin is the first time produced dentin, the oldest dentin in this thickness. It is mantle dentil, which then is outside the dentin. Then mm, here. There are the types of dentin. It is primary physiologic dentin, secondary physiologic dentin, and tertiary dentin, which is reparative dentin or reactionary dentin or irregular secondary dentin. And this primary physiologic dentin is divided into mantle dentin, circumpulpal dentin, peritubular dentin, and intertubular dentin. Let's discuss this mantle dentin, which is the first outer layer of dentin in the crown and differs from the bulk of the circumpalpal dentin in four features, this mantle dentin. It is slightly less mineralized here. Then the collagen fibers are largely oriented perpendicular to the animal dentin junction. For this reason, it can be distinguished from the circumpalpal dentin beneath using polarized light. And this dentin, the bulk of the dentin, is called the circumpalpal dentin beneath the mantle dentin. Yes? Then, uh, here, what we can say, this is sigmoid, this is primary curvature of dentin, and primary dentin is the dentin which is laid down to give the tooth its basic shape, and it after tooth has erupted and the root is completed, there produced the dentin is called the secondary dentin. Before eruption, the dentin which is laid down before eruption, it is primary physiologic dentin, but after eruption, this and root formation completion, this dentin is called secondary dentin. Yes, this mantle layer you can see here. This is primary dentin, and let's see, this is a mantle layer. Mantle layer varies in width from 20 micrometer to 150 micrometer. The special properties of mantle dentin prevent small cracks developing in the enamel near the junction from spreading into the dentin. Here, the three-dimensional scalloped architecture of the enamel dentin junction and the extension of some dentinal tubules into the enamel, you can see here, and you can already know that they are spindles, enamel spindles, which were described with enamel. Then the mantle dentin, these tubules in dentinal tubules branch profusely in this region, and it undergoes mineralization in the presence of matrix vesicles. And here you can see this is mantle dentin, and when you view in polarized light, mantle dentin is red, and this is circumpalpal dentin, and this is enamel, and mantle dentin differs from circumpalpal dentin, and maybe the reason is their perpendicular location to the dentin enamel junction. And here you can see this red line and red part, which is a mantle dentin, and you can see how these dentinal processes are branched in the mantle dentin. Then, uh, now you can see the here there is interglobular dentin. Between the mantle and circumpalpal layers is a layer of dentin which is calcified but not entirely, not fully calcified. And this layer is called interglobular dentin in globular layer. And this is globular layer and interglobular layer. Much of the mineral in dentin is deposited as globules or chalcospherides. Here you can see these are chalcospherides, And these in most areas fuse, as these calcospheroids fuse uniformly, and they calcify this tissue. And this is called mineralization front, this appearance of calcospheroids and their union. In small areas, usually beneath the mantle layer in the crown, and beneath the granular layer in the root, there are some artifacts, some abnormalities, and these are called interglobular dentin. And here you can see that it is hypomineralized. This dentin is and the mineralization and fusion may be incomplete when ground section are viewed in a transmitted light. The internal reflection is such appearance. They have such appearance, and it is thought that these spheres are uncalcified interglobular areas in here, and it is high magnification. But you can notice that the tubules, dentinal tubules, pass without deviation through interglobular areas, and as interglobular areas remain uncalcified, peritubular dentin is also absent from the tubules as they pass through the interglobular dentin. And the such interglobular such layer you can see in the let me yes, yeah, I show you in the root here. And in the root, this is called granular layer. In ground section, the periphery of the dentin is the root. In the root is marked by the presence of a dark granular zone, and it is called granular layer. Various explanations for this appearance have been suggested, and the most currently accepted is that the dentinal tubules in this area branch more profusely. Here you can see they have these tubules, they have a tree shape appearance, and they branch more profusely, creating air spaces in ground sections, and this result in reflection of transmitted light. And difference in the rate of formation of coronal and reticular dentin could explain why this appearance is seen in the root but not in the crown. And here this appearance, these three shape tubules, they are stain filled tubules and view in thick sections, and they have such three top appearance, somewhat supporting this idea that they are filled with stain. This granular layer is hypomineralized in comparison with the circumpalpal layer dentin, but this may be the result of the presence more tubular branches. And there is also the near this layer between cementum and this granular layer, there is such glass appearance layer, which is called hyaline layer in the root. And this layer may be, it is a hyaline layer may serve to bond cementum to the dentin, and may be of considerable clinical significance when considered periodontal ligament. Then the bark of the dentin is circumpalpal dentin, as you know, circumpalpal dentin here, the bark of the dentin. These all are parts, types of primary dentin prior to eruption. We discuss mantle dentin, we discuss interglobular dentin, which is at the periphery of mantle dentin. We discuss peritubular dentin, intertubular dentin, and circumpalpal dentin, which is such appearance. Let's see, circumpalpal dentin. No, we have no. Now, let's speak about predentin. Predentin, you can see how this predentin differs from dentin. Here you can see this is predentin and dentin, and they are different in appearance, in color. And it is dentin, predentin is the distinct pale staining. It has pale staining appearance, and it reflects the difference in the composition of its matrix and dentinal matrix, and the mineralized circumpalpal dentin, other circumpalpal dentin. The mineralizing front may show a globular appearance. You can see this globular appearance of this predentin, reflecting the mineralization process. The predentin is the initially laid down dentin matrix before it mineralization. It is hypomineralized. It is not mineralized. During mineralization, the matrix undergoes considerable modification, and principal role of the odontoblast process in predentin is the secretion of matrix components. In mineralizing dentin, the role of the odontoblast process is to participate in the modification of the matrix and perhaps also in the mineralization. Predentin width of predentin layer may from 10 to 40 micrometers, depending on the rate of which dentin is being deposited. And here on this layer you can see this is dental pulp, and odontoblasts are a layer around the dental pulp layer. And do you hear me?
Yes, yes, we can hear you. We can
Yes.
hear you.
I changed, but do you see the presentation?
Yes, yes, we can
Yes.
hear.
And these odontoblast layers surround the dental pulp. It is external in the periphery, and they produce. You can see on this layer these odontoblasts and their processes in tubules, and these processes are propagate into the whole thickness of dentin. You know that dentin is produced whole time and throughout the life. Now, here you can see the difference between predentin. This is freshly secreted layer of unmineralized dentin matrix, and it always lies on the formative surface of the dentin, and you can see these odontoblast cells, and you can see the propagation of their processes. Here, this is granular layer in the root, and they are called granular layer of tomes. And this is hyaline layer near this granular layer, the next of this granular layer, and this layer is between cementum and granular layer of the root. We discussed granular layer, this clear hyaline, the width of hyaline layer 20 micrometer in wide, as yes. Now, you know, this is primary curvature. Sometimes this it is sigmoidal in crown and more less pronounced in root, and this is the primary curvature in the crown of primary dentin. And there are some structural lines in dentin, in sections of dentin. viewed by different techniques. A variety of lines approximately perpendicular to the dentinal tubules can be seen, and this description and explanation of these lines vary considerably because there are two related groups of lines, those originating from curvature in the dentinal tubules and those arising from the incremental deposition of dentin and its subsequent mineralization. The lines which are accompanied with the curvature are called the lines of Schreger. These are longitudinal lines which are seen only on longitudinal section, and this is they coincide, they are peaks of these curvatures, peaks. When they coincide, they give such appearance in the longitudinal sections, but on the horizontal sections they have such round appearance, and there is only one visible and maybe two visibles. Then the lines, also there are secondary curvature here. Sometimes these lines of secondary curvature, they coincide and give such appearance, which are called lines of Owen. When these secondary curvature coincide, they also give rise to an optical effect resulting in the appearance of lines of Owen. Here, these secondary curvature lines of Owen here and here, and they are unusual in primary dentin but are sometimes seen, and they exaggerated line are found in the border of primary and secondary dentin, and they between dentin formed before and that formed after birth. These lines are seen. There are also structural lines in dentin, which these lines arise from the incremental deposition, like animal. You know that there are short period lines, period lines, and long period lines. Short period lines are called von Ebner's lines, and long period lines are Anderson line. And here you can see these lines are perpendicular to the dentinal tubules. They form right angle to the dentinal tubules. And these short period lines, they coincide, they are circadian lines. Circadian lines is in animal, and they reflect the circadian rhythm of dentin deposition in 24 hours in daily formative processes. And here you can see these are perpendicular to the processes, perpendicular and small lines, and the distance between these two lines may be 4 or 5 micrometer in length, and they are seen as bending along the dentinal tubules. And there are also the long period lines, and these long period lines contain about 6 or 10 pairs of short period lines, and they appear 6 or 10 days periodicity, and such periodicity is unknown, but they coincide in animal long period lines, and maybe some periodical hours are in the body. Um, this periodicity coincides, and you can see these are they are here. You can see they are these horizontal lines, and they contain 6 or 10 short period lines. Then know that dentin is laid down throughout the all life, and the dentin undergoes age changing. Once the tooth is erupted and fully formed, dentin can undergo changes that are either related to age or occur as a response to a stimulus applied to the tooth, such as caries or attrition. Yes, and these changes after tooth is erupted, has erupted, after root is completely developed, such dentin which is formed is called secondary dentin, and the secondary dentin is physiologic dentin. It is made from such odontos, the same type of cells as odontoblasts, odontoblasts, and its structure is very similar to that of primary dentin, and it may be difficult to distinguish between these two, between these two. Primary and secondary dentin are often delineated. Here you can see this. Here you can see the line, the boundary between primary and secondary dentin. This secondary dentin, here you can see it encircles the dental pulp from outside. Here you can see this dentin, this boundary between these two type of dentin, and they are often delineated as a result of a change in direction of the dentinal tubules with coincidence of secondary curvatures. This produces a particularly pronounced contour between this type of dentin. The same odontoblasts continue to lay down similar dentin, and the tubules of primary and secondary dentin are continuous. They may be continuous in these two type of dentins. The increased crowding of odontoblasts as secondary dentin formation continues throughout the life, and the slower rate of deposition make the tubules patterns of secondary dentin a little less regular than that of primary dentin, and the incremental marking somewhat closer together. Secondary dentin formation begins at the completion of root formation. After root formation is completed, secondary dentin formation begins, and the main coincidence would seem to be the apparent completion of root formation as secondary dentin still forms in unerupted infected tooth. Secondary dentin forms most here rapidly. Here you can see this is primary dentin, this is secondary dentin, and you can see the boundary between these two types of dentin. And here you can see this primary dentin, and this is the curvature between these dentin. Mhm. How they change their direction, it is indicated, matched by the arrowheads, and this is this delimits the junction between primary and secondary dentin. Secondary dentin forms most rapidly on the pulpal floor. Its continuing deposition leads to smaller pulp chamber and narrower root canals in older patients. In physiological aging, especially in root dentin, the tubules can become completely occluded with peritubular dentin to form translucent dentin. Where they are occluded with peritubular dentin, their refractive index is similar of intertubular dentin's refractive index. And when you put this such tooth into the water, it seems translucent. Here on this image, you can see that this is secondary dentin and primary dentin, and the change of orientation, and this gives the boundary between two types of dentin. And you can see the cells, odontoblasts, which give rise to secondary dentin. And also you can see the predentin, which is unmineralized dentin, and it has the different color than the mature and mineralized dentin. Now, about tertiary dentin. Tertiary dentin is very often called as reparative dentin, or this is secondary dentin at the periphery of dental pulp. You can see this is secondary dentin, and the tertiary dentin appears after trauma, after infection, after they are called also reparative dentin, or they are called also reactionary dentin, and this describes their function in dentinal, in this tertiary dentinal structure. This is primary physiologic dentin. Here in the surrounding the pulp is secondary physiologic dentin. These two types of dentin are formed by odontoblast, but the formation of tertiary dentin is not fully understood because there are the different type of cells, and they react to the stimuli, outside stimuli, and the stimuli of different types of extent may be applied to teeth at different stages of development of or aging, resulting in a response tissue that vary considerably in appearance and composition. It may resemble, here you can see, it may resemble secondary dentin in having a regular tubular structure. It may have a few or irregular arranged tubules, or maybe relatively atubular. And here you can see that these tubules from primary dentin, they are continued into the tertiary dentin, and here you can see this part is tertiary dentin in response to deep caries, and mm, tubules between normal dentin and tertiary dentin will therefore be lost in many cases. Then uh, this response from tertiary dentin, thereby of their response in many cases, it is called as reparative dentin and reactionary dentin. You know that the pulp does not seem to respond to stimuli by increasing the rate of deposition of secondary dentin, but by inducing previously quiescent odontoblast-like cells. It is thought that there are the cells which are odontoblast-like cells. It is the more likely explanation that there are odontoblasts. Like, you know that we discuss it on next lecture, that pulp contains a lot of cells such as odontoblasts, also odontoblast like cells, or there are stem cells which can transform into the odontoblast like cells. Also, dental pulp contains a lot of immune cells such as macrophages, fibroblasts. You know that this dental pulp is connective tissue. It contains fibroblasts, macrophages, dendritic cells, also it may contain the plasma cells and other all immune system cells. And these cells can also be found in the dentinal tubules beside the nerve endings, beside nerve odontoblast processes. There may be the processes of such, for example, dendritic cells or other antigen presenting cells. And this tertiary dentin is produced by odontoblast-like cells. They are the newly differentiated cells responsible for tertiary dentin formation and are very similar to odontoblast in that they produce type 1 collagen and dentin sialoprotein, which is specific protein for bone and dentin, and dentin specific proteins. This is more likely explanation that these odontoblast-like cells are differentiated from stem cell population in the absence of any epithelial contribution. You know that this odontoblast differentiation is due to the uh, epithelial mesenchymal interaction and growth factors. They cause this is reciprocal interaction. When predentin, for example, cause production of enamel, and then enamel production cause enamel dentin production and mineralization. This is called reciprocal interaction. And in this case, there is no this reciprocal interaction. And it is question how these stem cells can differentiate into the odontoblast-like cells. Yes, and it can be envisaged that the appropriate bioactive molecules are necessary for such differentiation, such as cytokines, growth factors, and locally synthesized and released these factors which are produced as the response to inflammation or other external processes or caries or other, and they send the stimulus such as dental caries, for example. They send the stimulus to this dental pulp, and these affect several factors have been shown to induce the formation of repair dentin in the pulp of experimental animals. And these stimuli which are sent from caries, from other degeneration, other trauma, they can cause the differentiation of stem cells in dental pulp. And they can induce the formation of repair dentin in the pulp of experimental animals. Yes, such experiments. When they were injected native dentin or crude extracts of dentin matrix, fibronectin products, and bone morphogenic proteins, it might be envisaged that the addition of suitable activating substances in the region of pulp exposure might aid and speed clinically the formation of repair dentin over pulp exposure. Now, let's summarize the primary dentin and the secondary dentin. They are physiologic dentin and they are formed by odontoblasts. These are cells which are differentiated in the dental pulp and they produce dentin. Yes, and this is physiologic process, but in tertiary dentin you can see this is dentin and this is tertiary dentin, tertiary dentin, and it looks otherwise than dentin. Yes, and This tertiary dentin, it is the response to outer, to external stimuli, such as trauma, such as caries, such as, thereby it is called reparative dentin, reactionary dentin, because its lay down is reaction to the external factors. It's connected to external factors. Then, sclerotic dentin. You know that with the aging, the peritubular dentin is filling with aging, is filling these tubules, and this is the physiological response to aging. Physiological response, you can see this, forming a translucent dentin. These dentinal tubules are occluded, they are entirely occluded. Dentinal tubules commonly fill in as a response to an external stimulus, such as under slowly advanced caries or beneath areas of severe attrition, and this type of dentin is termed sclerotic dentin. And it's like translucent dentin will present as areas that lack structure and appear translucent. Here you can see. Little is known about precipitated material, but it appears to differ from peritubular dentin, and it does so not to be formed by the odontoblast. The mineral is crystalline and possibly an apatite. Although plate-like crystals of octacalcium phosphate have been seen in this sclerotic dentin, the mineral concentration is significant higher in sclerotic dentin than in young dentin, consistent with the closure of the tubal lumens. Then their apatite crystals are slightly smaller in sclerotic dentin. The elastic properties of dentin are unchanged by transparency. Sclerotic dentin, unlike normal dentin, exhibits almost no yielding before failure. In addition, the fracture toughness is lowered roughly about 20% in sclerotic dentin. In tubules exposed by attrition or caries, some occluding components may be derived from saliva at the same time. Yes, and you can see here, this is sclerotic. You can see that this dentin is entirely occluded, and the sclerotic dentin's amount increases with age, and believe that the mechanism to protect pulp. And here you can see this is the intertubular dentin. You can see this crystal, and sometimes these are open, patent, and sometimes they are occluded, almost occluded. And here you can see the sclerotic casts in the tubules here and here, and they blocked these dentinal tubules. Yes. Then, what is it? There are, yeah, you can see that here, there, you can see that this is the sigmoid, this is primary curvature, which begins from pulpal surface and ends at the dentino enamel junction. this line. And here on the left, you can see the at higher magnification, smaller deviations, and this is ground sections when the tubules, they are obliterated in the primary odontoblasts if the primary odontoblasts are killed by an external stimulus or retract before peritubular dentin occludes the tubules, empty tubules will be left. There are empty tubules, and they can be sealed at their pulpal end by tertiary dentin. And when ground sections are prepared and mounted, the mounting medium will not enter these sealed off tubules, and they will remain air filled. And under light microscope, transmitted light will be totally internally reflected, and these tubules appear black. And here you can see this is black, and they are called dead tracks. Tubules which are affected by caries may fill with bacteria, and they also have some such appearance. Also, odontoblast processes may disintegrate or retract, and these space which stay after their retraction, they also is filled with air, and it has such black appearance. And these empty spaces behind these processes after retraction, they have such appearance. And these dead tracks can occur due to the death of odontoblast from crowding in pulpal holes. Yes, here you can see these black data tracks in here, and you know that they can be from they are air-filled spaces which ends are sealed. Now, you know that dentin is permeable, and the permeability of dentin is expressed in substances which can apply to its outer surface, they being able to reach and affect the dental pulp. Yes, and there are some it depends on several factors, such as the dentin surface is exposed by caries, attrition, abrasion, or trauma, and they are able to pass through the odontoblast layer, which present a barrier to molecules of higher molecular weight. But sometimes the drops can be appear on dentin, and this indicates that there is the internal pressure which exerts on the dentinal surface. Then the response to outside stimuli comes from the dental part, but it manifests the manifestation in the structure of the dentin is it produces the deposition of tertiary dentin provides a barrier to the progress of caries and the toxins, and the presence of secondary dentin and its continuing deposition throughout life, although not a response to external stimuli, contributes to the barrier function of the dentin. Thus, secondary dentin and tertiary dentin, they both serve as barrier, contributes to the barrier function of the dentin. You know that dentin is sensitive, and it is the question where these nerve endings. There was such hypothesis that dentin nerves are in dentin, but there are no arguments against this view, and there is no then it is not found the nerve endings in the dentin, and in addition the application of local anesthetics to the surface of dentin does not abolish the sensitivity. Then the odontoblast processes that they can serve as nerve endings, and there's then these processes can propagate this, conduct this pain to the central nervous system. But most plausible hypothesis to explain the transmission of sensory stimuli suggests that all effective stimuli applied to dentin caused fluid movement through the dentinal tubules, and that this movement is sufficient to depolarize nerve endings in the inner part of tubules at the pulpal predentin junction and in the subendothelial endoblastic neural plexus. Some, for example, some stimuli such as cold, osmotic pressure, or drying would tend to cause fluid movement outwards, while others, such as heat, would cause movement inwards. Movement in either direction would mechanically distort the terminals, and this cause the generation of depolarization wave, and then the propagation of nerve impulse. Then there sometimes there is hypersensitivity of dentin, and this process is defined as a short, sharp pain arising from exposed dentin in response to stimulus such as thermal, tactile, osmotic, and chemical that cannot be described to any other form of dental defect or pathology, such as dental caries or lateral pulp canals. Its incidence in the population varies between 10 to 20% and is most commonly associated with the buccal surface of canines and premolars, especially in teeth that show gingival recession. Such dentin has tubules that are patent. Exposed dentin in which the tubules are not patent is not sensitive, and sometimes dentin resorption takes place. You know that the bone resorption takes place and the remodeling, and in bone there are the special cells which are osteoclasts. You know that osteoblasts, they form the bone material matrix, and the osteoclasts are for resorption of bone material. And here, resorption in dentin is usually associated with inflammation. When the cause is unknown, this condition is termed idiopathic resorption. Dentin resorption may be initiated from two sides: from the pulpal surface, when it is known as internal dentin resorption, or from root surface when it is known as external dentin resorption. Now we'll finish the dentin. It was the huge material, yes? It is difficult for you. Are you here? Now, is it hard material for you? Hello? Hmm? Do you hear me?
Yes, yes, we hear
Yes.
you.
This is, we finished this description of dentin. And how do you think? It is hard, or I'll send on this weekend. I write down these questions on Dentin and send and
Okay.
upload on Drive. And
Okay.
the next lecture will discuss dental pulp.
Okay.
Yes, dentin and dental pulp together they form complex, but because it is very huge material, we separate it into dentin and dental pulp, and we describe dental pulp. And from these four topics, such as you can use my presentation, you can use the which topic. This is chapter 9 in Birkovich, chapter 9, Dentin. You can read from textbook, you can use my presentation, you can use my lecture, and I'll send the questions. And
Okay.
on next week we discuss it. And from the 11th of May, you maybe have this midterm one. Any
Okay.
questions? Any
No.
questions? Mm, then see you.
See you, Haj.
Yes.